This conference will now be recorded.
It's good when I remember to hit record. My name is Heather Tabasola. I'm the co-lead of the seminar series with Jens Nilsson, and this seminar is part of NOAA's EcoFOCI biannual seminar series focused on the ecosystems of the North Pacific Ocean, Bering Sea, and U.S. Arctic to improve our understanding of ecosystem dynamics and applications of that understanding to the management of living marine resources. Today's talk is the last of our spring seminar series. We sincerely thank you for joining us this season. Our fall speaker lineup will be found via the One NOAA Science Seminar Series and on the NOAA PMEL calendar events. We anticipate the series to start in November. We have not confirmed the date as of yet, so just please stay tuned and watch those two avenues just to stay up to date. Right now, please just double check that your microphones are muted and you are not using video. During the talk, please feel free to type your questions into the chat and we'll address those at the end of the talk. So today's speaker is Calder Atta, and he joins us to discuss how he is using a comprehensive genome-wide data set to address a complex history of disagreement between flatfish studies that have spanned more than a century. He is a graduate student at the University of Washington in the Tornabene Lab and a research assistant in the UW Ichthyology collection. His primary work in the collection involves facilitating the transferal and archival of larvae and egg specimens from NOAA's annual Alaskan ichthyoplankton surveys. And this talk is actually part of his first chapter of his master's thesis. I was also reading some behind what Calder had put on Luke's Tornabene's website for his master's as part of the group. And happy Earth Day, everybody. And since it's Earth Day, Calder had put this quote and in his bio, and I just thought it really resonated for today. So Calder, I'm sorry for calling out on this, but it was really great. So Calder wrote, "I believe that every organism has a story to tell that teaches us to appreciate the beauty of this finite planet and about our role in understanding and protecting its invaluable resources." And with that, I turn it over to Calder.
Thank you, Heather, for that wonderful introduction. As Heather mentioned, today I'm going to be presenting on some of the work that I've been doing in Luke's lab. And a lot of what we do focuses on biodiversity and phylogenetics and taxonomy. And so I kind of wanted to get us started by just asking a very basic question: what is a flatfish? Now, many of you are probably familiar with flatfishes, but when I think about the morphology of what is a flatfish, these three main characteristics come to mind. They have asymmetrical eyes, so they have eyes on one side of their head. They have an extremely compressed body and they lie sideways, usually near on the seafloor. And they also have these very long dorsal and anal fins that run the entire length of the side of their body. And that really helps them get more surface area to help them propel themselves through the water. The flatfishes make up an order called Pleuronectiforms, and this is a very, very diverse group with 816 species, and it's still growing. So with such a unique body plan and lifestyle, we would expect that all existing species would share a common ancestor that also had these traits. But there's actually been a lot of debate to whether that's actually true. So this is a phylogeny that was published fairly recently by Shi et al. 2018. And this is one example where of several studies that have actually suggested that there are two origins of the flatfish body plan. So is this true, or is this an artifact of our methodology? This debate has been going on for a while. It's been suggested almost a century ago, and then it really picked up steam about 8 years ago. And to this day, it's still not a well-resolved question. There's not a whole lot of consensus on it. And this problem in the flatfish phylogeny is only one issue, and the flatfish phylogeny actually has a large history of many problems that have often not gone talked about. And so how did we get here? So like we briefly touched upon, the flatfish, the history of the flatfish classification and phylogeny has gone on for well more than a century. And to understand it, we kind of have to go back to the beginning. So early in the 1800s, when people were sort of just putting together the flatfish classification, we didn't have as many species of flatfishes, so all of them were sort of put into one family, Pleuronectidae. But then over the years, many species got discovered. And then it's kind of hard to, people started realizing it was hard to place all of this diversity into one family. So they created more families. So they restricted Pleuronectidae to only flatfishes that people typically called flounders. And they made a new family for the soles, and then a new family for, what are they called, the spiny turbots. But this wasn't the end. Of course, new species keep getting discovered. And they're like, well, okay, we have to do this again. And so with the flounders, they decided to split them up by flounders that were right-eyed, so had eyes on the right side of their head, or flounders that were left-eyed, which were in the family Bothidae. So that's all well and good until you get more species. And then over the years, they kind of decided to throw that whole model out the window. Having eyes on the right side or the left side of your head is very easy to see for a human, but maybe it's not the most evolutionary relevant characteristic. So now they broke up the Pleuronectidae into all of these families, and now we have these families for the Bothidae, and then the soles got split up, and now we have about 16 families now. So most of that was using morphological characters, looking at things that look the same as other things and grouping together based on morphology. But eventually, people started using more quantitative tools and using phylogenetics and to try and untangle this phylogeny. But even with phylogenetics, you run into a lot of problems. The reason this group keeps getting broken up into smaller and smaller pieces is that with every new phylogeny that gets published, there seems to be conflicting results with past phylogenies. So to understand that, we kind of have to think about how phylogenies are made. So this is sort of an example of how historically phylogenies have been made. So on the left, you have a matrix of usually presence-absence characters that are morphological data, and then you use a computer to generate a tree that is the most likely to produce the results of your data set. So ideally, from the data set that you give the computer, the collective information that we would get from these characters would result in one well-supported phylogeny. But in reality, this is rarely what happens, and especially in the flatfishes, there's an extremely high number of trees that are very likely to fit the data. So one way to decrease biases from any one character trait is to use more traits. So instead of using the morphological traits in this table, we could instead use genetic sequences. So that's what a lot of people have been doing for the last several decades. This is just an example of a few of the most comprehensive data sets that people have done in the last few years. And basically what I want you to take away from this is that there's a big disparity in how much genetic material is used and how many genes are used. And this is somewhat problematic because if different studies are giving different results, that could stem from them using different genes, because genes evolve at different rates and are under different selective pressures. So your results are going to depend on what genes you select. So one solution to this problem is, once again, just to get more genetic data by sampling from more genes. The ultimate solution to that would be to get full genomes for all of for all of your species, but that's not very feasible. Doing this for many species would be very expensive, very time consuming, and you would probably break a computer trying to make a tree with all of that information. So instead, what we've been doing, or what we did for this, was use a method called Exon Capture. So Exon Capture is a method that was developed by our collaborators in Shanghai, China. And the idea is that we've already mapped the genes in a lot of model organisms, like zebrafish and tilapia. So we can use these data sets to try and find the ones that are also present in the organisms that you want to study. So the benefits of this, they've already tested this or used this to work out relatively recently evolved species and also deeper relationships. So it's kind of in that scope of evolution that we're working with. So the set of genes that we're using comes out of this publication, Zhang et al., 2018. And in this study, they provide a data set of 4,434 exons that should work for any ray fin fish. So these are all single copy nuclear genes. The size range is pretty wide. Typically, they're around 250 base pairs, but the largest is, as you can see, is up to 5,000 base pairs. And the total genetic material that's in this is over 1 million base pairs. So if we add this to the previous graph, we can see that in comparison to the previous studies, this is a lot more data that we're dealing with. It's about 100 times more genetic material. However, this method is fairly time consuming, so we're limited in the amount of species that we could include. So this is by species, so not the highest, but we did manage to get 89 species out of 816, and we tried to get, we made sure to cover most of the major groups, so we got 11 families out of 16. Our samples came from several fish collections, so the University of Washington, University of Kansas, Louisiana State University, CIRO in Australia, and a few samples also came from here, AFSC. So once we have the sequence data, there's a pretty extensive computational work pipeline that we did to prepare the data before making trees. And because of the uncertainty sort of surrounding the flatfishes, we wanted to try several different tree construction methods. So these were the two main methods we did, the concatenation method. So in this method, all the genes are sort of stitched together into one long sequence per sample. Then the tree is constructed from this master gene sequence. And then on the right, with the gene trees method, we created one tree per individual gene, and then we combined all of those gene trees into one species tree. And then after this, the last thing we did was we did a test to find genes that were most clock-like, meaning they appear to have mutated in a linear fashion over time. And so in theory, this should give you more accurate results. But I'll show you how that came out. And then with our filtered data set of clock-like genes, we then applied both methods once again. So that gives us 4 trees in total. And here are the results. So one of the aims going into this was that by inferring from more data, we would see some improvement in clarity on what the true phylogeny looks like, but that didn't really happen. While the trees sort of superficially look similar, they actually differ quite a lot in terms of how the families are related to one another, which has also been a recurring problem in previous studies. So what this is showing that even with the same data, the result can differ based on the method that you use to construct a tree. Like in previous studies, we kind of found that many of the relationships are still unclear. So where does that leave us? Where do we go from here? So I went back and reviewed previous studies again, and it turns out there's some consistency about what is unclear. So I decided to write down some guidelines on how we should be tackling this problem in the future. So first of all, we've been trying to tackle the issue of limited data by increasing the amount of genetic material being used. But even at this scale, different genes give us different results. And if we think about it, that might not actually be a big problem. The data is just data, and it's just in the case of the phylogeny, sometimes there are rapid radiations that create species very quickly. And in these cases, I think we should be asking ourselves, how important is it to parse out the details of every speciation event? How likely are we able to find meaningful results, and how much would it cost? So in phylogenetics, if we can't figure out who's more closely related to who, we can collapse branches into polytomies, and I'll show you what that looks like on the next slide. Besides using more sequence data, one way to gain information is to gain more information is to sample from a greater number of species. But as said before, it's kind of not feasible to sample from a large amount of data, and especially from 816 species. So to work around this, we can infer relationships at a very broad level, which is sort of what everyone's been doing so far. But we can also focus in on specific groups and do more comprehensive studies and focus on where there are knowledge gaps. In order to find the knowledge gaps and the polytomies that are sort of created from these radiation events and are unlikely to be resolved soon, I used our trees and corroborated our data with previous phylogenies. This was a pretty intense challenge since the literature on flatfish phylogenetics and classification goes back so far into the 1800s. And so basically, this is the result of combining our results, or the results of many papers with phylogenies of the flatfishes. So this is a tree showing how all the families are related within the order Pleuronectiformes. So there are two main radiation events that I point out in this tree. So the first one, the one on the left, this one is at the very base of the tree, and this is the event that's making us wonder if the flatfish body plan arose multiple times. And then the second one, the polytomy in the middle of the tree, has 5 different lineages coming out. And looking at the phylogenies across publications, there's very little consensus on what's really happening in this part of the tree. So both of these areas, I think, are going to not be very likely to be resolved anytime soon. And then if we sort of zoom in to the family levels, I also have family level trees. So these are the phylogenies, some of them to genus, some of them to species, if they're well studied, to the best of our knowledge so far. The Pleuronectidae on the right is a very well established group at this point, and that probably doesn't need much more analysis in the way of species relationships, but I did find a lot of families that still needed comprehensive work. So these 8 families, really are data gaps in the flatfish phylogeny. There needs to be a comprehensive phylogenetic analysis with all species or nearly all species to sort of resolve or report on those relationships. I've also compiled a list of genera where
cautiously take it.
I've also compiled a list of genera where one or more studies have suggested that they could be invalid. So I would say all of these genera need to be re-examined at some point. And then the last list I have are genera that have never been included in any molecular phylogeny. So we don't know where they go. We know they're within their respective families, but we have no idea where they go on the tree within those families. And my hope going forward is that we can use this information to guide us on where we need to focus our attention. And then you don't have to tackle each of these groups individually. If you did a comprehensive analysis of the Bothidae, you could include all of the genera for the Bothidae and sort of knock those out at the same time. So for me personally, for my future plans, I'm working with our collaborators, and we are compiling a database of Exon Capture data. So it's not just flatfishes, but with all fishes. So our data is going to be part of that data set for future studies. The other potential thing we could do is GGI, or gene genealogy interrogation. And this is a technique to sort of look at individual genes to see how much each one is contributing to the final tree and if there's potential bias. So that can sort of tell us where what's causing some of the specific patterns that are conflicting with each other. And then with the trees that I produced, I was going to do time calibration. So time calibration is basically mapping the phylogeny onto a geological timeline. And we do this typically with fossils. And so you use the fossils to put a date on certain points on the tree and sort of scale it to real geological time. And then once the tree is time calibrated, we can ask more interesting questions about how flatfishes evolved and moved spatially through time. And so this is sort of hinting at chapter 2 of my thesis, which is looking at Alaskan flatfishes, how certain Alaskan flatfish species have evolved in the North Pacific. So I think with that, I'll start taking questions.
Thank you so much, Calder. That was really good. I clap for everybody because I know it's weird probably giving a talk and not hearing that. All right, questions, please type them into the chat, and Jens and I will monitor that and facilitate that with Calder. And again, just a reminder too, this is the last of the EcoFOCI talks this season, so we will see everybody in the fall, and dates for that will come out later in the summer. Calder, oh, here we go. Okay, I was gonna say, you stymied everyone. So here is a question from DF. Any way to filter the data set to improve the signal to noise ratio?
So filtering noise, so one way we could do that would be the clock-like filtering. Basically, by selecting genes that are sort of, I guess, that seem to be accumulating mutations more evenly. That's the goal, is to reduce the noise and remove genes that are sort of behaving more radically. I'm not sure if that completely answered your question, but yeah.
Thank you, Calder. People are slow to type in questions today, so I know it's a rainy day in Seattle, so maybe everybody's just still asleep.
Maybe.
I had no idea how many times like the phylum, like how many different classification groups, like how many times that has been moved through. That it doesn't surprise me, but it was fun to learn about that.
Yeah, it was fun for me to dig through all of that.
Yeah, was it? Was it really fun? I liked how you laid it out on the slides. Okay, Cosmin, do species from the same family encounter similar environmental variables?
Ooh, so on a family level, it differs widely. Let me go back to this picture. So these are the 16 families. So just by looking at how many species are in it, you can sort of get a grasp. More species are going to have a bigger, overall family-wide geographic range. Some species, so the Pleuronectidae, which is probably what most of what we are familiar with, those are primarily in the northern temperate and polar northern hemisphere. And so typically with that, you're going to see there's still going to be a variety of substrates that they're going to be on, whether it's like more silt versus more rocky. But a lot of these are so there are temperate and polar species. The Atrorapsetidae with the 4 species are only known from Antarctica. There are a lot of species that are tropical too. So the Bothidae, the left-eyed flounders, and the Rhombosulidae are more temperate Australia. But when you get into the tropics, you're spanning the global tropics. That's a lot of area. And you're in the Pleuronectidae, you're dealing with species that are going down as deep as 1,000 meters. So there's a huge variety in terms of environmental variables as well. Yeah.
Thanks, Calder. Colleen says, thank you for the great talk, Calder. I'm excited to see the step with the fossils. Where do you plan to get the fossils from? UW collection or beyond?
So there aren't very many fossils right now. Right now I have a list of 24 fossils, so I wouldn't physically be handling the fossils. But basically I get this would be another dig through literature, dig through what other people have used for fossils. One thing you can do for time calibration is also use other people's trees. So basically you just have to put a timestamp on a node and then how you want that node to be restricted to that timestamp. So basically the process would be going through the literature, finding a publication that describes a fossil and then what time period it was from, and then placing that on somewhere on the tree. And by using multiple timestamps, you can sort of calibrate it. It's not the easiest process. There's definitely a lot of error associated with it. One challenge is that you need to have I'm doing air quotes, accurate tree. So something I have to decide is what I think the best tree sort of to use would be for that. But yeah, I wish I were handling the fossils. That would be really cool.
Thank you, Calder. Thank you for the question, Colleen. CJ, I see your question. I'm actually going to save that one for last. I'm going to jump back to it. Question from Melanie. With the families that need further analyses, among those, are they particular species relative? Wait, I don't know if I read this right. Okay, wait. With the families that need further analysis, among those families, are they particular species relative to the other better resolved families?
It's a bit of a mix. So the 3 biggest families, just looking at the numbers, the Bothidae, the Solidae, and the Cynoglossidae definitely all need thorough re-examination. There doesn't seem to be too much of a pattern in terms of which ones were studied. The Pleuronectidae has been studied a lot just because it was that first family that included all of the flatfishes, and as it sort of accumulated all these things based on arbitrary, well, somewhat arbitrary, this thing looks like a flounder. It accumulated a lot of like the classification accumulated a lot of structure that was misleading based on the morphologies that people were using. So once genetic techniques picked up, it really people were sort of hammering it out, trying to figure out the Pleuronectidae. I think some of the groups that are sort of hard to come by, the Etrapsetidae, the Antarctic ones. No one's ever looked at the interrelationships of those species, although it's just 4 species. But I mean, it's a lot of work. If I go back to this slide, on the bottom, I'm showing you sort of the numbers of so the families that need comprehensive analysis are 8 out of 16. So half of the group, and of course 3 of those have over 100 species. And then looking at the 2 right columns, so just general with problems that need to be addressed, we're looking at 45 out of 127. So it's a lot. A lot of work needs to be done. And I wish it were easier to collaborate with people, because this is such a large group and they're global, right? So we have to look at different collections, especially groups that are harder to find and in remote areas are going to be harder to get a hold of. Yeah.
Thanks, Calder. Thanks for the question, Melanie. Morgan just had a statement. He said, I really like your comment that there is some consistency about what is unclear. That is the fundamental reason for the study of ichthyology, and I would probably add the fundamental reason study for most science. So I'm going to jump back to CJ's question here, if nobody else has other questions. And CJ's question is, how could you use what you've learned from a systematics perspective to help inform fisheries management?
So this is sort of the taxonomy response to fisheries management. A lot of these species, especially in the Pleuronectidae, and so if we want to talk about Alaska, I think there are 23 species of Pleuronectidae there, and there's also Paralichthidae, I think, as well. So for species, and a lot of those species are commercially harvested, and relating it to how this affects fisheries, I would say that taxonomy, you need to have taxonomy as the foundation for any sort of getting any sort of ecological information. So for example, if you had a population that you thought was one species and you thought it was doing well, but then you find out by doing population genetics that there are 2 populations, and then even more so if they're 2 species, your numbers are going to completely change. If it's not one continuous population, then how you interpret population size is going to differ dramatically. And so getting all this figured out provides like the foundation for all other ecological questions. I hope that answers. I hope that's a sufficient answer.
DJ does say good answer and good talk. Thanks, Calder. All right, if there are no other questions, Calder, is there anything last minute that you want to add?
This was the first time I've given a remote presentation, and it was an interesting experience.
Yeah, I think you did a great job. I know it's weird not seeing people, but
yeah,
people do tend to be, they are engaged. All right, so Calder, again, thank you so much for joining us. Thank you for closing out our season. And to everybody else, we'll see you in the fall. Thanks again.